Brain-computer interfaces refers to a system that allows users to interact with a device using solely brain activity. Although brain-computer interfaces is a widely researched field in human-computer interaction and a promising alternative to traditional methods like keyboard that uses physical touch or voice commands, it is rarely used on the healthy population as a method of interfacing with devices. For example, some applications of brain-computer interfaces include speller or movement controls of a robotic arm, which are specifically for people who have permanent disability like loss of limb or paralysis. The reason why most healthy people are not keen to use brain-computer interfaces is due to limitations like discomfort, low usability and low information transfer rate. In this research project, we will be focusing on research on the characteristics of steady-state visually evoked potentials under different situations in hopes to improve for future applications. A typical BCI system is separated into three segments. They are mainly the hardware, software and interface segments. The hardware segment is responsible for sensing the biological activities in the brain, which forms the key component of the interaction or communication between the user and the device. The interface segment refers to the program or application software that the user directly interacts with. Usually this segment consists of instructions and stimuli to evoke the users to produce recognizable brain signals which will be received as input to the system. Lastly, the software segment refers to a backend analytical algorithm or tools that could include machine learning or statistical analysis to classify the signals that were received previously. It is very difficult to identify what type of signals the users are producing and its relations to the user's intention manually, so this segment is also very important for the BCI application to work accurately. Steady-state visually evoked potentials is one of the most popular methods in brain-computer interfaces and also the method that we will be studying in our research experiment. It refers to a type of brain response generated involuntarily when a person looks at a flickering visual stimulus. The response is uniquely identifiable by the frequency of flicker, hence, people may use it as a speller, where each alphabet is associated with a specific flickering frequency. In this method, electroencephalogram is used to capture brain activity of the user as electrical signals. Steady-state visually evoked potentials can be identified in these signals using machine learning classification or statistical analysis. The specific goal of this project is to discover new characteristics of steady-state visually evoked potentials-based brain-computer interfaces by identifying the relationship between spatial attention, viewing angle, and stimulus interference with respect to performance in terms of classification accuracy and information transfer rate. The results might contribute to discovering more practical ways to design a brain-computer interface. For example, studying the spatial attention relationship allow us to evaluate if it is feasible to use peripheral view-based brain-computer interfaces, and understanding viewing angles helps us consider the optimal distance for placing visual stimuli. The findings from this research experiment are also going to be published on the International Conference of Engineering in Medicine and Biological Society. To study the effects of spatial attention and stimulus interference, the experiment was designed such that research participants have to place their chin on top of a chin rest so that their head would not move about during the experiment. Additionally, a desktop eye tracker is used to keep track of where the participants are looking at during the experiment so that we can confirm if they're using peripheral vision during covert attention tests. In terms of the actual experiment, participants have to complete the task of transmitting information by looking at flickering visual stimuli that will evoke a reaction in the brain. We created single and multiple stimuli experiments using both overt and covert attention to compare the characteristics of different settings. The figure in the middle illustrates what participants will see during single stimulus experiment, and the right figure shows the same for multiple stimuli.
So start ready? Right? So this is the actual experiment. You
develop using a CyclePy library.
Develop using the CyclePy library, Python 3. So this is the screen. Before the experiment start, right, there will be some instructions to tell the users where to look at. So this is experiment 3, covert attention. So users are supposed to focus their attention at the center and not at the visual stimulant.
Can?
Yep.
Okay.
Okay.
Explain.
So this is how the experiment sequence will look like in the actual experiment itself. The user will look at the center for covert attention, and he will try to focus his attention on the target stimulant, which is directed by the arrow. So there's a red boundary to tell the user where to focus his attention at also. So using this,
yes,
we'll try to see if the SSVEP signals are higher if the users are focusing their attention on specific targets.
Okay,
yep.
That's all right?
Yep.
The results from analysis of the collected data suggested two things. Firstly, visual viewing angle is not a significant factor in the performance of SSVEP detection accuracy, as seen from the figure on the left where the coefficients values are similar across all visual angles. Secondly, in covert spatial attention settings, lower stimulus frequency correlates to higher detection accuracy, as seen from the second figure on the right.